The second step in RNA processing is a process known as RNA splicing. As we previously said, the purpose of the RNA splicing is to remove non-coding parts of RNA known as introns, so that the only RNA left is mature RNA made up of only exons. RNA splicing begins as several snRNPs, or small nuclear ribonucleic proteins, bind to the ends of each intron on the pre-mRNA. Just as it is finished being transcribed, and each snRNP has an RNA component called the snRNA. At the boundaries between the introns and exons are consensus sequences, which are essentially short stretches of DNA that appear with very little variation in many different genes. These consensus sequences are similar to the poly-A tail in that a certain sequence in the snRNP will bind to the consensus sequence through complementary base pairing. Once the snRNPs have located the boundaries of the intron, the actual splicing begins. One snRNP binds to the pre-mRNA at the 5' exon intron boundary, as another binds to the pre-mRNA at the 3' intron exon boundary. Once the snRNPs are in place, proteins are added using energy from ATP, which forms a large RNA protein complex called a spliceosome. Each spliceosome is composed of 5 small nuclear RNAs, or snRNAs, and a range of associated protein factors. These snRNAs combine with the snRNPs to then form the spliceosome. The snRNAs that make up the major spliceosome are named U1, U2, U4, U5, and U6, and participate in several RNA-protein interactions. The spliceosome then cuts the pre-mRNA at the designated boundaries. Then the ends of the exons join together, thus creating mature mRNA.